Hi everyone, in this video we're going to be looking at transport across the cell membrane. The key words that we'll be focusing on are selectively permeable, passive transport, ATP, diffusion, osmosis, osmotic pressure, facilitated diffusion, isotonic, hypotonic and hypertonic. So the learning objectives that we're focusing on today are to describe diffusion and osmosis, and we're going to explain how a concentration gradient can influence those two types of transport. Before we start this lesson, it's a really good idea to have read pages 34 to 38 of your textbook. If you haven't, please stop the video and do so, or come back to it straight after. So if we look here at a cartoon picture of the cell membrane, we can see all the different constituents that we identified last time. So we have cholesterol here, we have proteins, we have the phospholipid molecules, and we have the carbohydrate parts. Now, just as a reminder, when we look at these phospholipid molecules, we understand that the head, which is chiefly phosphate, has a charge and is polar. The tails, which are made of, are fatty acids. These are lipid and they do not have a charge. So these are classed as non-polar. Now we know that the phospholipid bilayer, that's these two layers here, orientate themselves in this way with the tails pointing inwards because they are hydrophobic or water-fearing, whereas the heads are hydrophilic or water-loving. So why is that important? Why do we need to know about the property of a membrane? Well, it really dictates what can enter and exit a cell. So for example, small hydrophobic molecules, water-fearing molecules, they can actually get through the tiny gaps in that plasma membrane. So we're talking about really small things like gases such as oxygen and carbon dioxide. Now these are described as lipid soluble because they can get past these tails without being repelled. Next type of molecule we're going to be looking at are another group of smaller molecules such as water, urea, which is a waste product, and glycerol. These are non-polar, so they're not charged, and they can pass through, again, through the gaps in that membrane, or they'll pass through a protein channel. Next, we're going to be looking at large molecules. So these are non-polar, which means that they really find it hard to get through these gaps. They can't do it, they're too big. So they require help from proteins. So that needs to be a channel protein, which we're seeing here, or a carrier protein, which I'll show you later on. So if we summarize that using this diagram from your textbook, we can see that from the phospholipid bilayer, there are some molecules such as water and smaller molecules and ions that will pass through a protein channel, so they need a bit of help there. Large molecules just can't pass through at all, hence why the membrane is selectively permeable. But other things, smaller things like gases like oxygen and carbon dioxide, they can diffuse easily through that membrane, while alcohol, steroids and other lipid or fat soluble molecules can easily pass through. Now, when we start talking about transport across the cell membrane, we can break this up into two categories. So in your reading, you might come across different terms for the selectively permeable membrane. It might be described as semi-permeable, which means some things can come through, or differentially permeable, and that means it differentiates on the molecules. So all of these different terms, all they mean is that they allow certain molecules to pass through but prevent others. Now the two types of transport can be categorized as either passive transport, which means that no energy is required, so that's really important, it just simply passes through without the need for energy, or it's active transport. An active transport is when we do require energy, and remembering that we use energy in the form of this molecule down here, adenosine triphosphate, also known as ATP. This lesson will just focus purely on passive transport. So the first one we're going to be looking at is diffusion. So in this animation you can see blue dye swirling around in water. Now you know from your own experience, if you leave for example tea in a cup with a tea bag, that tea will start to spread out throughout that water, or indeed if it's a coloured liquid such as cordial, that will spread throughout the water as well. Now why does this happen? Well, it is diffusion. Now this is a term that you absolutely need to know, and when you write down this definition, I need you to be using the key words in red. So diffusion is a passive form of transport, so it doesn't require energy, and diffusion only works for gases or liquids, and it's only when those substances move from a high concentration to a lower concentration until that they are evenly spread out. So in this cartoon diagram here, you can see that we have a high concentration of red molecules, and they're passing through that selectively or differentially permeable membrane, and they're moving from high concentration on this side to a lower concentration on this side on the right. Now over time we will end up with an even distribution of those red molecules on either side. Now it's important to note that these molecules are not static. That means they're not standing still. They are constantly moving, so while you have some molecules on this side, they may move across to the other side, but to make sure that everything is evenly spread out, some molecules from this side will move back across. Okay, so understanding that diffusion is the movement of molecules in a liquid or a gas. It's passive, so no energy is required, and we are moving from an area of high concentration to an area of lower concentration until the molecules are evenly spread out. Okay, so this little cartoon here is just to remind you that diffusion will take place much faster if you have a steeper or higher concentration gradient. It's a bit like being on a slide. If you have a slide that's really high up, you're going to go down that slide really quickly because you go from a high area to a low area. If that slide is has got less of a gradient on it, you won't go down as fast. You'll still move, but it will take much longer compared to this slide up here. And we can apply that logic to concentration gradients. If you have a really high concentration of particles on one side and hardly any on the other, these particles on the left are going to diffuse much faster to get to the other side and make everything even. However, down here, because we have a less steep concentration gradient, these particles will take a slower time, so they'll diffuse at a slower rate to get across. Okay, so this is a factor that affects diffusion rate, and that is the concentration. So quick summary here. Passive transport, so if you're passing something along, it takes no energy, so there's no ATP there, and it's going on the principle of molecules moving down the concentration gradient. So we say that they move down the concentration gradient from an area of high concentration to an area of lower concentration. Now this video here provides you with a summary of passive transport. I'll play that for you. Please make sure that you understand what's going on.
This means that some small molecules, such as oxygen and water, can diffuse across freely. Carbon dioxide and amino acids can also diffuse into or out of the cell. The net movement of molecules depends upon their concentration inside versus outside the cell. Molecules move naturally from an area of high concentration to to an area of low concentration. When molecules move in this way, no energy is required, and they are said to move by passive transport.
Okay, so just a recap of simple diffusion. It's the movement of molecules, as you can see here, from an area of high concentration to an area of lower concentration until they are evenly spread out. So in terms of the cell membrane, molecules that will move across the cell membrane through the little gaps in the phospholipids would be oxygen, carbon dioxide, alcohol and steroids, because they can pass through the lipid tails. Molecules that also move by diffusion but can't easily pass through those phospholipid molecules require channel proteins. These would be ions like sodium, Na for short, that's the chemical symbol, or potassium, which we give the letter K, or water. Okay, so by this point you should be able to complete these 3 questions. Now I've highlighted in red these command words. Now these are words that you'll see in your exam. So state what is meant by the term passive. So when we say state what the term passive means, you only have to give a simple, concise, 1-sentence answer. We're not looking for anything more than that. So that's a fairly low order recall question. Going up a notch, we're looking at describe questions. So you have to describe the term selectively permeable membrane with respect to the cell membrane. So that means that you're going to have to write more than 1 sentence and describe what we have learned so far. The third question is using the word define. So you have to give me a definition of the term diffusion. Now these questions are very common and you should expect to see something like that in your first assessment. Okay, so now that you've done those questions, we're going to be looking at another form of passive movement, passive transport, another type of diffusion, which is osmosis. Now, osmosis is the special diffusion of water, so you have to really get into your head here that osmosis is diffusion of water. Definition of that is that osmosis is the diffusion of water molecules through that selectively permeable membrane from an area of high water concentration to an area of lower water concentration. So in this cartoon here, I've represented that dotted line as our selectively permeable membrane. On the left hand side here, we've got pure water. And on the right hand side we've got solvent molecules, so that could be anything like sugar or salt. So let's just say that that's a sugar molecule. So these solvent molecules, these are sugar dissolved in water. When we look at the next picture over here, we can see that the volume of water on the left has gone down because of osmosis. Water has moved from an area of high water concentration to an area of lower water concentration. So the volume on the right has gone up because there are now more water molecules. So what we have on the right is more water molecules in the same volume. Now, the reason why we talk about more water molecules in the same volume is because this relates to osmotic pressure. The term osmo, that means water. So when we talked about osmosis, that was diffusion of water. So osmotic pressure is referring to pressure with water. So if a membrane is present, water will always flow from a high water concentration to a low water concentration, that's osmosis, and it can change the osmotic pressure of cells in your body. So osmotic pressure is the pressure created by water moving across a membrane due to osmosis. In this cartoon here, we can see that water is flowing from a high water concentration to a lower water concentration. Again, these pink circles are representing our solute. So by comparison, because this has more solute molecules in it, it will have less water, hence why osmosis occurs from left to right. The reason why we have a higher osmotic pressure where there's more solute is because water is flowing from high to low water concentration, and because we have more molecules in a small space, it's increasing the osmotic pressure. Okay, so here's just a summary of what osmosis is. It's the diffusion of water moving from a high water concentration to a lower water concentration. Another way we can say that, and you can sometimes see this written in assessments, is you're going from low solute to high solute concentration. So take your time to look at this diagram and understand that if we have a liquid with a low sugar or solute concentration, then it must have a high water concentration. And the converse is true here. If we have lots of sugar or a high solute concentration, the water concentration will be lower by comparison. If you can understand this, you'll be able to work out questions regarding osmosis. Okay, so the next thing we're going to be looking at is something called tonicity in cells. And what this really means is the volume or concentration of water surrounding a cell. So remember, the word extracellular means outside of the cell. So this is fluid outside of the cell and its effect on cells. Now, the reason why we want to study this and why it's important is because the human body likes to be in balance. You know, on a hot day, if you are losing water, you don't feel great, and if you have too much water, then you have the urge to get rid of it through urination. There are three key words in relation to tonicity that we need to know about, and these are isotonic, hypotonic, and hypertonic, and we're going to talk about each of those in turn. The first one we're going to be looking at is isotonic, and you need to know that the term iso means equal. So what we're really saying in an isotonic solution is that it has an equal concentration of water inside and outside of a cell. So this cartoon here is showing a red blood cell, and we can say that the solution around the outside this red blood cell has an equal volume of water as the inside of that red blood cell. So the liquid that makes up your blood is called plasma. And we have lots of cells that are bathed in plasma, such as red blood cells. So if your blood plasma is isotonic, as it should be, then you won't have any water leaving your cells and you won't have too much water going in. You will have this nice even net flow of in and out to keep everything balanced. Okay, the next one we're going to be looking at are hypotonic solutions. Hypotonic solutions mean low solute concentration, which effectively means high water concentration, because if you don't have very much solute, sugar or salt for example, you're going to have lots of water. Now, that spells trouble for your blood plasma. If your blood plasma is really watery, if it doesn't have very much solute in it, it's going to have a high water concentration. Now, if we look at this cartoon over here, we can see that that means water is going to move from a high water concentration outside the cell to a lower water concentration inside the cell by osmosis, and that causes your red blood cells to burst. Now, the reason why red blood cells burst is because they don't have a cell wall the way plant cells do, so they will just keep filling up with water and eventually rupture like a balloon skin. We definitely don't want that to happen, and we don't want our red blood cells to burst, because remember, they do a really important job of transferring oxygen around the body. The opposite of a hypotonic solution is a hypertonic solution. So hypo means high solute concentration, or the converse of that would be a low water concentration. Now again, remember that osmosis is the diffusion of water from a high to low water concentration. If you have blood plasma that is very sugary, so let's say for example you've had a very high sugar meal and the blood plasma concentration is too high in sugar, it's not having enough water to balance it out. So again, if we look at this cartoon over here, we can see that the surrounding solution of this red blood cell is low water concentration. That means the inside has a higher water concentration, therefore water will leave the cell by osmosis. Now because water is leaving the cell by osmosis, it causes them to shrivel up. Now again, that's not a great place to be because your red blood cells have a biconcave donut type shape, and that helps them to transport oxygen around the body. So if you're changing that shape, you could be altering that red blood cell's capacity to transfer oxygen around the body. Okay, so at this point, you should be able to answer these questions. First question, identify the types of solution in beakers A, B, and C. So we're going back to those words regarding tonicity: hypertonic, hypotonic, or isotonic. For each one of these three beakers, you should be able to state which direction osmosis is occurring in. Is it going to be in, out, or both? You should also be able to identify the solution with the highest water concentration relative to the red blood cell. So when we say relative, we mean in comparison. So these three questions are basic 1-mark recall questions. You shouldn't be writing any more than one sentence for those. And for the final question, it's an explain question. We would call this an level A type question, or an A grade question. So you have to put a bit more thought into it and use your key terminology. And you need to explain why blood plasma must be isotonic to red blood cells. So pause the video here, see if you can answer those questions and complete page 20 of your booklet. Next, we're going to be looking at facilitated transport. This is still passive transport, so no energy is required, but this time we are using proteins. We can use channel proteins, as you can see in the middle diagram here. So it just forms a pore where these molecules can pass through from high concentration to low concentration. Or we can use carrier proteins. And this is, again, passive transport from high to low concentration, but the protein changes shape. Now, the reason why we have these channel proteins and carrier proteins is because some molecules are unable to get across the membrane because they are hydrophilic, and that means that they are water-loving, and the tails are hydrophobic, and they will repel them. So examples that you should know about that require channel and carrier proteins are water, glucose, and some key ions such as sodium and potassium, which we've mentioned before. So we're going to look at the example of glucose. Glucose uses facilitated transport, and it does it in quite a unique way. You can see in this cartoon here that our glucose molecule binds to our carrier protein, and when it does that, it causes the carrier protein to change shape. And when it changes shape, it opens at the other side and it drops that glucose molecule off on the inside of the cell. So remember, we need glucose for energy. So this would be the extracellular environment. Glucose binds to that carrier protein, the carrier protein changes shape and opens up, transporting glucose across that membrane to the intracellular environment. Okay, and again, this is a passive form of transport. This video here just summarizes facilitated diffusion. Please make sure you watch and you listen carefully.
Molecules with strong electrical charges, such as ions, cannot simply diffuse across the cell membrane. No matter how small the ions are, their charge prevents them from moving through the layer of lipid tails in the middle of the membrane. Many molecules, including proteins, are simply too large to diffuse across the cell membrane. Carbohydrates, such as sugars and starches, are also too large to diffuse across. Large molecules are transported across the membrane by carrier proteins in the membrane. Glucose, a sugar, is one molecule transported in this way. Glucose attaches to the carrier protein. The protein changes shape and releases the glucose molecule inside the cell. In this way, glucose is moved down its concentration gradient from the area of higher concentration to the area of lower concentration. Like diffusion, this process, called facilitated transport, requires no energy. It is.
Okay, so we reached the end of this lesson, and here is a summary of key facts that you should know. First of all, passive transport does not require energy to move substances across the membrane. Diffusion is the movement of molecules in a liquid or gas from an area of high concentration to an area of lower concentration until those molecules are evenly spread out. Osmosis is a special diffusion of water, and molecules can move across the membrane using protein channels and carrier proteins by passing through layers in the phospholipid bilayer. The rate of diffusion is faster if there's a greater difference between the two sides, so if there is a higher concentration gradient. Solutions that have substances dissolved in them, solutes dissolved in them, can be described as isotonic, and this means that the solution and the cell have equal water and solute concentration. They can be described as hypertonic, which means that they have a lower water concentration and a higher solute concentration than the cell. Or they can be hypotonic, which means they have a low solute concentration but a higher water concentration than the cell. Facilitated diffusion is a form of passive transport that uses protein channels or carrier proteins to passively move molecules across the membrane, and that means without energy. Your task by the end of this lesson is to complete the work on pages 16 and 20 of your class booklet.